Welcome back. So, in this class we are going to take a very beautiful example of biophotonics, very beautiful example of light interacting with biological matter, light interacting with tissues, and we will finish this module of interaction of light with biological matter. So, let us discuss photoprocesses in biopolymers. So, when we talk about photoprocesses in biopolymers, we are mostly discussing that there is a chromophore that upon absorption of light initiates a set of reaction, initiate a set of events that goes through. So, I think I discussed a bit about chromophore, but if you are confused about the term chromophore, let me define it. Chromophore is basically an organic compound that absorbs light. That is the, so if you have a chain of molecules, say protein and nucleotide, not every part will be absorbing light, or not every part will be absorbing, say, optical frequency of light, UV or visible or IR. Chromophore is that specific molecule, the specific molecule of a large compound that actually absorbs a specific wavelength of light, and this chromophore, the work of the chromophore is in determining the overall color of the entire molecule, right. The entire molecule, the entire compound made up of molecule A, molecule B, molecule C, that is complicated compound. A specific part of it absorbs light. Some part is absorbed, some part is reflected, some part may get fluoresced, and this chromophore, this specific molecule is the one of a long chain of molecule which is responsible for absorbing light and thereby producing the color either by emission or by reflection. So, chromophore is the light sensitive part of the compound, the light sensitive molecule which actually is directly affected, and rest of the part of the molecule of a long complicated chain is especially like in say in proteins. Proteins have huge amount of amino acids, not all of them will be absorbing light, but if a protein is connected with a chromophore, you will see soon, that part absorbs light and that subsequently starts a chain of events. So, chromophore is a specific molecule, an organic molecule that absorbs light and give rise to color, give rise to other subsequent processes. Chromophore usually can exist either just by themselves or a part of a long chain of molecule. So, the processes that the chromophore, that absorption of light in a biopolymer, they have these characteristics. They are highly efficient, very fast. The primary steps leads to series of complex steps, and the large number of intermediate steps are also formed by a variety of physical and chemical processes. So, light absorption by chromophore not only give rise to a chemical process, but also we will see physical processes, right. Physical processes. Remember Frank Condon principle, when you absorb light, the atom swells up because the electron has moved to other level. That electron then overlap the electron of other atom in a molecule, then there will be a repulsion, then there will be an intermediate change in the distance R1 versus R2. Come on, I have discussed this Frank Condon principle. So, there is a physical change that takes place or can take place when a molecule absorbs light, a part of a molecule absorbs light. Now, the subsequent steps are formed in dark and hence called dark reaction, meaning that once the light has hit that molecule and a chain reaction have had started, you may or may not require the light to continue. The light can be switched off. The light can be switched off. Whatever the chromophore need to do, it has already done, and now the domino effect have started. Now, the domino effect have started. Like in a domino effect, you just click or you just flick one domino, you do not need to do it all the time, you just flip it once and everything starts falling, all those dominoes starts falling. Similarly, here the light needs to hit, the reaction start, and one after another reaction keeps on happening, and therefore, since they do not require light, we call them dark reactions. The important thing here is that this photo-induced process is cyclic, meaning in the absence of light and give it some time, it will return back to the original position what you have started with, right. So, you have a molecule, you have shown some light, you have hit it with a light or you have hit it with a photon, the chromophore has absorbed and it has started subsequent chain reaction. The chain reaction has gone on, the light has been switched off, but after some time this reaction reverses back to the original position where the chromophore is again ready for another set of light, another set of photon to work on it. So, it is a cyclic process, it returns back. The primary example, the most beautiful example that I would like to discuss with you today, remember I discussed a bit about it, I mentioned a bit about it in the introduction video, is human eye and vision. This is my favorite topic and every single day I read, I learn something new from it. Vision, how beautiful it is, and this makes all our world go round. We see this beautiful world and it's actually a miraculous process or a very, very intelligent process that that fascinates me on a daily basis. So, this is your eye from outside, and if you look into it, this is the internal structure. So, you have a clear cornea where the light basically falls. You have a diaphragm like an opening which is called pupil, you know, the pupil opens and closes like a diaphragm of a camera. You understand diaphragm, previous old camera there used to be this kind of a enclosure which opens up and closes up. So, our eye exactly has this pupil which fixes the amount of light, fixes the amount of photon that can enter through the cornea, and that amount of photon, that amount of light falls onto the lens. Several times this lens, this internal lens that is present in our body are malfunctioning, or they do not work, or their focal point have had changed, and that is primary reason why we require an artificial lens, basically a spectacle. But nevertheless, let us continue. So, light coming from this clear cornea gets, you know, little bit, it falls through the pupil and falls onto the lens. The lens, like anything, focuses the light into the back of your eye, which contains this mushy tissue, the retina. Every one of you know or heard of retina. This retina is the tissue which contains photoreceptor cells, and in today's class, the specific example of tissue light interaction, I am going to discuss about light reaction with retina or retinal tissue. I told you I will be giving a specific example of light interacting with tissue. Thus far we have discussed general interaction of light with tissues and cells and molecules. Today I will be discussing specifically light interaction with retina. Okay. This retina is then connected with the optic nerve. So, the retina is the transducer. So, basically three processes are happening in vision. Think about it. You have reception. You have reception of light. Not all of light can pass through. You have pupil to take care, you have lens to take care. So, through the cornea, light enters, the pupil somehow controls it a bit, the lens focuses it. So, all this part is reception of light. This light then falls onto or focuses by the lens onto the mushy tissue which is retina. In retina, the transduction takes place, where we will see in a minute that light signals through photoreceptors, through cells which are sensitive to light, get converted into electrochemical signals. So, that is the transduction part, where one signal is converted into another signal. Then that electrochemical signal is taken through optic nerve to our brain, visual cortex in the brain, where the coding takes place, where these electrochemical signals are converted into perception or converted into vision. and it's fascinating. It's like current flowing through and going into a processor. The processor is processing the signal. This is what I'm referring to the electronics engineers and other engineers. You will know this is 10 times more sophisticated than this, and we take it for granted. Let me take you step by step. So what exactly happens? So the human retina. The human retina contains primarily 3 types of cells. The human retina, the mushy tissue that you saw at the back of our eye, contains primarily 3 types of cells. The first one, the first layer, contains rods and cone cells. All of you have heard about rods and cone cells, but today I am going to show you a very, very cool picture of rods and cone cells. So rods and cone cells consist of approximately 100 to 120 million rods and 78 million cones. Rod cells are the one which are incredibly sensitive to light. Rod cells are the one which are incredibly sensitive to light but not to color. They gives you shades of gray, brightness, shades of gray, black and white and gray and all of these things. Very, very minute differences in gray can also be picked up by our human eye because of the presence of rod cells. Cones are very less sensitive compared to rod cells, but they provide color vision at higher level of illumination. So we have more number of rod cells, less number of cone cells. Rods are for brightness, shades and gray, cones are for color. Rods are highly sensitive, cones are less sensitive. Very little amount of light can give rise to shades of gray in rod cells, can result rod cell to do its thing. Cone cells on the other hand require a little bit higher level of illumination. So these are the rods and cone cells. Okay. Then there are these bipolar neurons or bipolar cells that are basically intermediaries that connects these rod cells and cone cells with ganglion cells. Okay, these are the intermediary, they are the interconnects. Electronics engineers or electrical engineer knows why we require interconnects in electronic circuits. So bipolar neurons or bipolar cells are the interconnects that simply transmit data, information from rod cell to ganglion cell. The ganglion cell is the one which is part of the neuron or the part of the visual information processing, which conveys the visual information as encoded by the retina via the optic nerve to the brain. So ganglionic cell is where is the extent up to which your brain is coming into effect. Okay. So this is the reach of the brain. Well, brain reaches everywhere, but you know what I mean. So cone cells are the photoreceptive cells where the actual light reception is taking place, where light energy is getting converted into some kind of electrochemical energy, and that electrical impulse or that electrochemical impulse is being carried forward by our ganglionic cells via the bipolar neurons onto our brain, where we are perceiving what we are seeing. There is a difference: perception and seeing. This part is seeing, that part, the brain part, is perception. So let's try to understand a bit more about the human retina. So this is the cool picture I found from Encyclopedia Britannica. This you should look at. So this is the structure of the rod cells and cone cells. Immediately you will know why they are called rod and cone. So because it looks like rod and cone, and it has large amount of mitochondria. Why do we require mitochondria again? I forgot. Can you remind me? There are nucleus, but most important features of rods and cone cells are these flat pancake-like structures, flat disc-like structure, CDs-like structure, these disc-like structure one after another. So you have these pancakes, they call it lamella or whatsoever. I call them pancakes. I don't know what you are going to call, discs-like structures, and they have the central fibril and well, life science students will be telling me how I have mispronounced or how this is important, but look at how beautifully these two cells are, and these are the one which is allowing you to see the world, see my video. So these are the rod cells and cone cells, and let's understand a bit more about the science of vision. So you see these lamellas, you see this disc. These disc contains a protein called rhodopsin. These disc contains a protein called rhodopsin. Actually, you will be able to tell me that it's a pigment. The protein is opsin. Yes, I was watching cricket and I would probably mispronounce it as off spin, like the spin bowling, but it opsin. Yeah, opsin. That's the protein, and it is connected with a chromophore, which is retinal. Let's understand this. This is very, very interesting. So, you have a chromophore material, an organic material that absorbs light, and that is called a retinal. This is that retinal, and with this aldehyde bond CHO, it is connected with a protein called opsin. Retinal is the chromophore, opsin is the protein. Combination together, we call them, the total thing, rhodopsin. We call this together rhodopsin. This is the chromophore. This is the chromophore retinal, or in particular case we will call it cis-retinal, 11 cis-retinal. You know from chemistry the difference of isomerism, cis and trans. Usually cis is where the same types of atoms are in the same side, and trans is where same types of atoms are in opposite side. Cis and trans are isomerism. Isomerism means the molecule is the same molecule but different arrangement. So, it is the same molecule, exactly same thing. We see it in organic compound having all the time. So, the same arrangement, molecule A and molecule B are arranged like this in cis, molecule A and molecule B are arranged like this in trans. These are isomers. So, the retinal chromophore is an isomer. So, this chromophore is connected with the protein opsin, and it forms overall the rhodopsin. So, rhodopsin 11 cis retinal, that is the chromophore, is covalently bonded to the protein opsin, opsin, not off spin. This retinal forms the chemical basis of animal vision, but that is not the important part. I am itching, I am actually anxious to teach you the most important part of vision. So, ready. You have a retinal chromophore connected with the protein, right? This is the retinal, this is the chromophore, this is the part which absorbs light, and this is connected with the protein and the entire thing. When a retinal absorbs light, the retinal has a structure like this, the retinal has a structure like this, you can see here, the cis retinal has a structure like this. Upon absorption of light, there is a change in the shape of the retinal molecule. From being cis, it has become trans. So, this upon absorption of light becomes straight. Previously, no light, the molecule is this, you have light hitting it, the shape of retinal is changing. The shape of retinal is changing upon light. Why the shape change? Go back to Frank Condon principle, but upon getting light, upon getting illuminated by light, the retinal molecule goes into a different shape, isomerism forms. From cis, it becomes trans. So, when there is a physical change happening in the molecule, this activates the opsin protein that it is connected with. It activates the opsin protein that is connected with. Previously, it is connected, but the thing that is connected with it, the opsin protein connected with it, is not activated. This energizing or this changing of the actual shape will activate the rhodopsin. What happens when this protein, the most important pigment or rhodopsin, gets activated? A chain reaction starts. Rhodopsin will activate another protein called transducin. Rhodopsin protein, so retinal absorbs light, its shape changes, changing of the shape gives a kick to the rhodopsin it is connected with, gives a nudge to the protein it is connected with. That proteins in turns give nudge to another protein which is transducin. The transducin produces an enzyme that breaks down this nucleotide, I think guanosine monophosphate. The transducin produces an enzyme that breaks down a nucleotide. You should know by this time what nucleotide is. This nucleotide breaks down the channel Na plus and Ca2 plus. These are the ion channels in the plasma membrane that it closes. So, again domino effect, one after another. Retinal, it absorbs light, it kicks rhodopsin. Rhodopsin kicks transducin, another protein. Transducin kicks, produces an enzyme that breaks down GMP. GMP produces a loss of Na plus and Ca2 plus channels in the plasma membrane of the rod cell. So, you have a rod cell. I remember the rod cells have membrane, any cell have a plasma membrane. The plasma membrane have gates that allows ion to come through. The gate is now closed. Sodium and calcium ions cannot go inside, positive ions cannot go inside the cell. Positive ion cannot go inside the rod cell. When positive ion cannot go inside the rod cell, what happens? When there is a positive ion formation outside and no positive ion formation inside, what happens? The inside becomes hyperpolarized or become more electronegative. Okay, you want to maintain a balance, you maintain a neutrality. Positive ions are no longer allowed to go inside the nucleus. So, you cannot neutralize it. You cannot neutralize it. There is a hyperpolarization. There is an accumulation of large amount of positive ions outside. So, by definition, there will be accumulation of negative ions inside. So, the membrane potential, the potential difference changes from minus 40 millivolt to minus 60 millivolt. Minus 60 millivolt in one single cell. You have 120 million of these cells in your body. So, when there is a potential difference, what happens? Electric current will flow. That current is then passes through the optical nerve onto your brain. Think about it. Let me repeat it once again. Retinal is the chromophore. This is this molecule connected with a protein. Upon absorption of light, this molecule hits the protein it is connected with. It kicks it, it gives a nudge. That is the rhodopsin protein. That rhodopsin protein kicks another protein which is transducin. The transducin produces enzyme that breaks down a nucleotide. The breaking down of the nucleotide results in closing of ion channels inside the rod cells and cone cells. When ion channels are closed, the potential changes. The potential change results in potential difference forming outside and inside the cell. When there is a potential difference, you know current will flow. That current is then transferred to our brain, where we are interpreting the current as visible information. Fascinating. Every day I see this and I think how beautiful this is happening. And think about it, minus 40 millivolt to minus 60 millivolt each cell carry this much amount of potential. This much amount is the potential difference per se. I know ophthalmologists or people who specialized in eye surgery or this ophthalmology, basically eye doctors, what we call them, they have to go through huge detail, and I am simply scraping the surface. This is incredibly superficial, but still this is incredibly fascinating. This is incredibly fascinating. A chain reaction is happening. Light is falling onto this molecule. The shape of the molecule is changing. There is a conformational, there is a physical change in this molecule. The physical change in the molecules kicks another protein. The protein kicks another protein. That protein produces enzyme that breaks down GMP. a nucleotide, the breaking down of the GMP results in blockage of sodium and calcium channels, ion channels in your cell membrane. Since sodium and calcium channel cannot go, since positive ions cannot go inside the cell become negative. If there is a difference in potential, negative inside, positive outside, current flows, and that current is interpreted, right. So, this is the overall process. This is the overall process where the this rhodopsin, the retinal part, upon absorption of photon changes its shape, changes its conformation, and this starts giving kick to other proteins, other intermediate proteins, and it results in change in electrical current. It results in change of electrical current, electrical potential within the cell, and that potential difference results in formation of a current flow. That current flow is perceived in our visual cortex as brain, as vision. Okay. Now, obviously, two things you need to know, that there are several intermediates upon light absorption. So, rhodopsins, when I am talking about the kicking part, one protein is kicking another protein, or one pigment is kicking another pigment. I have oversimplified things. There are several intermediate pigments, metarhodopsin, then bathorodopsin, luminrhodopsin. All these intermediate stages happens when rhodopsin gets activated by the retinal chromophore changing its shape, and all of these intermediates have different absorption maxima, and they produce all those shades of gray that we perceive. What happens when the light is removed or there is no light, i.e., when you close your eyes? When you close your eyes, it is dark. So, retinal goes back to its original position from trans to cis, since it has gone back from this to this, right. It is no longer kicking the rhodopsin. Rhodopsin is no longer kicking the transducin. The entire process is no longer carrying forward. You have rod cells with plasma membrane open for positive ion channels. So, there is no longer potential difference, or less amount of potential difference is present. It is no longer hyperpolarized or that much negative inside, and very little current flows. We call this dark current, and the cells are depolarized. Consider it like, electronics engineers consider it like the avalanche breakdown in your diode reverse bias. Remember diode reverse bias condition? No current flows or minority charge carrier flows up till reverse bias, but once you reach the breakdown point, huge amount of current flows. So, you close your eyes, the retinal is not functioning, very little current is flowing, dark current is flowing, consider it as minority charge carriers. Electronics engineers will be chuckling. Minority charge carriers are flowing, and you are not seeing anything or very little shades of gray. As soon as you open it, light enters, your rod cells get activated, the retinal changing its conformation, changing its shape from kink to this, and all the intermediate processes or the subsequent processes have started, and you start seeing things. How soon do you think that happens? How soon or how quickly your brain can start interpreting light? Okay? You close your eyes and open it. How soon do you think your brain start interpreting the light? So, understand the process, the fastness of the process. Is it picosecond, nanosecond, millisecond, microsecond? How much time it takes for you to open up your eye and see the world around, or the light to actually hit and your brain to work through it, the brain to process through it? How fast this process is? Are all processes this fast? Is your hearing this fast? Or say your touch or pain, for example, does it slowly grow or is it instantaneously like your eyes? Think about it. I especially have bulging big eyes, so I utilize them to give the example, and I know what you are thinking about or what you have thought about when you have looked at my lectures, and no, I don't. So anyways, read a bit about this part. This is fascinating, this fascinates me. But another important thing that electronics engineer would see, or you should invest a bit more time, is all those electrical impulses that goes out because of the electronegativity, because of the hyperpolarizability of the rod cells. How are these electrical current, because of this hyperpolarizability, is being interpreted in our brain? Is it same like the processor of your computer, where all those bits are going and your processor is churning and giving some kind of information? Can you replicate it? Can you make an artificial eye? Think about it. You have an electric current, you have an electronic circuit, okay? It is changing its polarization. Because of the change in polarization, the potential difference changes. Because of the potential difference changes, different amount of current is passing through. Since different amount of current is passing through, you get different information at the central processing unit, and the central processing unit is interpreting. That interpretation part, that electrical signal interpreting part, unfortunate as it is not part of biophotonics. Biophotonics deals with light interacting with it. That's the electrical, neural circuitry and that kind of subject that will digress. But if there is an interested electrical or electronics engineer who would like to investigate this one particularly, I strongly encourage you to look into it, see how our brain interprets signals, and these are nothing but electrical signals. So for all intent and purpose, our brain is a hugely complicated electronic circuit, and if it is an electronic circuit, can we replicate it, or can we at least repair it or modify it? Think about it. So that basically brings me to the end of module number 4, interaction of light with biological matter. We discussed about human eye, rod and cone cells. Maybe I was too excited to explain you this. Hopefully you understood little bit. Otherwise, just go through vision per se and the visual process. And this is a fantastic book as always. So that ends my module 4. I will see you in next week, where I think we should start discussing about lasers. Thank you very much.